Assalamu alaikum and good morning everyone. My name is Tanvir Muhammad Farhan and today I will present my topic entitled Creon, Non-Halal Evaluation and Formulation Proposal for Halal Compliant Alternative. Creon is one of the most commonly prescribed pancreatic enzyme replacement medicines for patients who cannot digest food properly due to pancreatic insufficiency. Although it is highly effective clinically, it contains ingredients derived from pigs which makes it unsuitable for Muslims under normal circumstances. In this presentation, I will explain why Creon is considered non-halal, and I will discuss the Islamic and regulatory perspectives and finally propose a scientifically feasible halal alternative. First, let me introduce the product. Creon is a prescription pancreatic enzyme replacement therapy, also known as PERT. Its active ingredient is pancrelipase. Its active ingredient is pancrelipase, which contains three digestive enzymes: lipase for fat digestion, protease for protein digestion, and amylase for carbohydrate digestion. The medicine comes as delayed-release capsules containing enteric-coated microspheres so that the enzymes are protected from stomach acid and released in the small intestine. Creon is mainly prescribed for patients suffering from exocrine pancreatic insufficiency or EPI, where the pancreas cannot produce enough digestive enzymes naturally. Now let's understand the disease itself. Exocrine pancreatic insufficiency occurs when the pancreas fails to produce enough digestive enzymes. Common causes include chronic pancreatitis, cystic fibrosis, pancreatic cancer, or surgical removal of the pancreas. Because of the enzyme deficiency, patients cannot digest fats, proteins, or carbohydrate properly. As a result, they experience symptoms such as fatty stools or like abdominal discomfort. And some people also see weight loss or some people see malnutrition. And there's always deficiencies of fat-soluble vitamins like A, D, E, K. Pancreatic enzyme replacement therapy works by supplying digestive enzymes directly into the small intestines during meals, which allows the nutrients to be absorbed normally. Now we move to the halal issue. The main reason Creon is considered non-halal is its active ingredient. Pancrelipase is extracted directly from the pancreas of the pigs, collected from commercial slaughterhouse. According to Islamic law, pigs and porcine derivatives are classified as najas al-mughallaza, meaning severe impurity, and they are prohibited for consumption. In Malaysia, MAS 24242019, which is the Halal Pharmaceutical Guideline, clearly states that halal pharmaceutical products must not contain pigs or pig-derived ingredients. Therefore, Creon's active ingredient does not comply with halal standards. So, besides the active ingredient, there are other concerns. First is the capsule shell. Creon uses porcine gelatin, which is produced from pig skin and connective tissue. Many Islamic scholars agree that the manufacturing process does not completely transform the material into something pure, so it remains non-halal. The second concern is the manufacturing process. Since the enzymes are extracted from pig tissues, there is also a possibility of contamination during slaughterhouse processing and manufacturing. In addition, if equipment is shared with non-halal products without proper halal assurance, halal compliance cannot be guaranteed. So now discuss the regulatory and fiqh framework. Despite being non-halal, Creon can still be used under specific conditions. Islam recognizes the principle of darura or necessity. The principle states that prohibited substances may become temporarily permissible if they are necessary to save a health or a human life, and there is no suitable halal alternative. So there must be no halal alternative if we want to make this a necessity. Currently, many Muslim patients with severe pancreatic insufficiency rely on porcine pancrelipase because no fully equivalent halal product is widely available. However, once a safe and effective halal alternative is developed, the justifications of necessity would be no longer there. So let's see the proposed halal alternative. My proposed solution is to replace porcine enzymes with enzymes produced through microbial fermentation. Instead of using pig pancreas, lipase can be produced from microorganisms such as Rhizopus or oryzae or Aspergillus niger. Similarly, protease and amylase can be produced using Aspergillus or oryzae, which is a microorganism which is grown on plant-based fermentation media. This approach eliminates animal-derived materials and reduces the risk of animal-borne disease, which is fully compatible with halal requirements. Modern biotechnology already uses microbial fermentation extensively in pharmaceutical manufacturing, making this proposal scientifically realistic. So let's discuss the halal capsule technology. Besides changing the enzyme source, the capsule itself should also be modified. Instead of porcine gelatin capsules, we can use HPMC, which stands for hydroxypropyl methylcellulose. HPMC is produced from plant cellulose and is already widely used in halal and vegetarian medicines. The enzymes would still require an enteric coating so that they are protected from stomach acid because the stomach acid may digest them. Suitable coatings include plant-derived pectin or synthetic eudrigide polymers. We can use these coatings. All excipients and raw materials should also come from halal certified manufacturing facilities that comply with JAKIM or IFA and CA standards. So let's do a comparative analysis. This table summarizes the comparison between the existing Creon formulation and the proposed halal alternative. The current formulation uses enzymes from pig pancreas and porcine gelatin capsules. In contrast, the proposed product uses microbial enzymes and HPMC vegetable. The microbial enzymes also show broader pH stability while eliminating the possibility of animal viral contamination. Most importantly, the proposed formulation satisfies MS-2424-2019 halal pharmaceutical requirements while maintaining therapeutic effectiveness. Therefore, it provides both scientific and religious advantages. So now let's discuss the impact on patient care. Developing a halal alternative would provide many benefits. First, Muslim patients would no longer experience religious concerns about taking lifelong medication. Second one is patients are generally more willing to take medicines that align with their beliefs, which improves medication adherence. The product would also fulfill the concept of tayyiban, meaning not only halal but also safe, clean, and wholesome. Finally, this product would appeal not only to Muslims but also to vegetarians, vegans, or individuals with pork allergies, expanding its global market. So now let's discuss the challenges and roadmap. Although promising, several challenges remain. Microbial enzymes have different activities compared with porcine enzymes, so dosage conversion must be carefully optimized. Researchers also need to ensure the enzymes remain stable while passing through stomach acid. Clinical trials must demonstrate that the new formulation performs as effectively as Creon in improving fat absorption. Finally, the entire supply chain from fermentation media to packaging must remain halal certified to comply product integrity. These are important steps before commercialization. So now let's go to the conclusion. In conclusion, Creon is an effective medicine for pancreatic enzyme replacement, but it is considered non-halal because both its ingredient and capsule shell are derived from pigs. At present, its use may be justified only under Islamic principle of necessity. However, by replacing porcine enzymes with microbial fermentation products and using HPMC-based capsules, we can develop a halal-compliant alternative that meets both pharmaceutical standards and Islamic requirements. Thank you very much for your attention, and yeah, thank you. Assalamu alaikum.